Influenza A viruses are responsible not only for seasonal flu, but also lethal worldwide pandemics over the last century. When a virus infects a host, it triggers an immune response that is both necessary for fighting the virus, but which can also contribute to the virus's pathogenicity by inducing an inflammatory response. This host response is controlled by a complex network of signaling proteins and transcription factors. While some host response pathways have been characterized in great detail, we have yet to gain a holistic understanding of the host response network to different strains of virus, as well as among different host species. Understanding this network will provide crucial information for the development of antiviral therapeutics. Toward this goal, systems virology projects are generating an unprecedented amount of data measuring host response to multiple pathogens at multiple readouts, including the transcriptome, proteome, lipidome, as well as host virus interactome and siRNA screens that identify host genes important for viral replication in the host. An open challenge is to integrate these data to reveal information about the underlying host networks. But it's not straightforward to integrate these different data types, as they measure different molecules and also have different amounts of coverage. A major goal of large-scale data integration is to be able to identify key regulators of the host response that can be pursued in more detailed experimental studies. So to respond to this challenge, we have developed an integrative approach to reconstruct host response regulatory networks from time series measurements of mRNA and protein levels across a panel of different viruses. Specifically, we were motivated to be able to leverage the richness of mRNA level data as well as make use of sparser protein level data. Our approach consists of 3 stages. First, we use the full gene expression data to infer a regulatory module network, one species at a time, that links signaling proteins and transcription factors to modules of co-expressed and co-regulated genes. For this, we use MERLIN, an existing approach developed in our lab, that learns the modules and network de novo from the expression data. Next, we employ a structured sparsity-inducing regression approach to integrate proteins as additional regulators of the gene expression in each module identified in the previous step. Now, in the first 2 steps, we identified regulators based on statistical dependencies. But in order to get somewhat closer to a mechanistic regulatory program, we next integrate physical interaction networks to try to identify connections between the regulators. Doing so also enables us to integrate other types of host genes that are important for viral replication or that are known to interact with viral components. So specifically, we considered a data set that spanned 2 host systems, Calu3 cells, which are a human lung cell line, and mouse lungs. These cells were treated with 6 different influenza viruses, spanning a range of pathogenicities, where pathogenicity is quantified by MLD50, or the amount of virus required for 50% lethality in mice. In the first step of our approach, we infer a regulatory module network using just the mRNA expression data from one host system at a time. So we concatenated together all of the virus time courses for each host to generate a single expression data set. The input to the MERLIN method, which is what we used for network inference, is a gene expression matrix, shown here is our nice heat map, and a set of candidate regulators, which include transcription factors and signaling proteins. MERLIN infers a regulatory module network, which is by simultaneously inferring modules of co-expressed and co-regulated genes, as well as individualized regulatory programs for the genes. So the genes that are in a module together are encouraged to, but not required to have the same set of regulators. This figure shows the modules identified for our human data There are 41 of them, and each row in this figure gives the average expression level of that module across all of the different virus treatment samples. So each of these columns is a different time course. These modules give us, they provide a useful signature of patterns of host gene response. So for example, this module, where now the rows are individual genes, shows this diverging pattern where the low pathogenicity virus treatments induce these genes, whereas the high pathogenicity viruses show repression amongst these genes. Next, we use a structured sparsity-inducing regression approach to integrate the protein-level data with these mRNA-level modules. So we decided to focus on learning regulators at the module level because the protein data was much sparser, having fewer samples and fewer genes represented. Typically in lasso-based regulatory network inference, which is to identify small sets of regulators for individual genes, regulators are learned for individual genes. So this would be, these circles are protein level regulators, and these are genes. So typically you would learn per gene regulatory programs or regulatory programs for the average expression level of a gene. However, in our approach, we use a multitask Group LASSO approach where we want to identify regulators that are good predictors of all genes in the module. So that makes it a multitask approach. And the grouping structure over the regression coefficients is defined by all of the coefficients linking a single protein to all genes in the module. This framework has one parameter, lambda, which controls the weight on the sparsity penalty versus the loss in the objective function. We compared the group version, or MTG lasso, to the regular per gene lasso, and observed that as we vary lambda, MTG lasso identifies fewer non-zero regression weights and giving a sparser solution. And furthermore, the error of the two models on predicting held-aside expression values is basically the same. So we think that this suggests that the grouping structure gives us an interpretive benefit without gaining error. So we evaluated our regulatory module network using traditional enrichment analyses and by comparison to other available data sets. This figure shows 16 of our human modules that are enriched for some interesting gene sets, including in pink, we have influenza host factors identified by other screening or literature studies. In green, motifs for important immune response regulators and these arrows or these lines link to different modules. And then at the bottom, different immune response gene sets, including antiviral processes and inflammatory response. Now on the left, I'm starting to add in some of the top regulators predicted by our method for these modules. Now these regulators come from the mRNA level data, and we can add in the regulators identified at the protein level data as well. And the regulators that are filled in with pink here, several of them we note were independently identified for importance for influenza replication. Furthermore, we were able to perform an siRNA validation study on some of our regulators and identified some additional ones that showed importance for viral replication in our study as well. And these are marked with these yellow stars on the figure. Now I'll walk you through our complete approach using one module that we found particularly interesting. This module, its expression pattern is shown here, and each row is a gene. It's very strongly enriched for interferon-stimulated genes, which are an important part of the host immune response. And now I'll reveal the predicted regulators of this module. So this group is from the expression level data, and this group is from the protein level data. And here we see there are some complete time courses that are missing at the protein level. So just to note that. So the regulators of this module do include known regulators of host response, which is something that is exciting for us to see, like the JAK-STAT signaling pathway, where these Those regulators are marked with these blue circles. But they also bring in novel potential regulators, some of which came up in our validation study as new host factors. So these two are marked with yellow stars. So our regulatory network so far links regulators to targets based on statistical dependencies. But in order to get somewhat closer to a mechanistic understanding of the regulatory program, we integrate sparse physical interaction networks. So the input to this data set of the approach, we do this on a per module basis, is a set of regulators identified by our previous methods, as well as additional host factors that are known to interact with influenza proteins. We also assemble a background network, for example, from BioGrid. One naive approach is to just overlay the regulators on the network and extract all short paths between them. But this will often produce something that's pretty uninterpretable, like this one. And it will generally include false positive interactions. So therefore, we used an integer linear programming approach to identify a smaller, sparse subnetwork that connects the regulators using just the minimal number of intermediates. This picture illustrates our predicted physical regulatory module network for that module we were talking about before, where the input regulators inferred by our method are shown with dark outlines. We have transcription factors down here at the bottom, and signaling proteins and protein-level regulators predicted near the top. We also encouraged our method to include known host virus interactions. So the viral components are marked with these yellow hexagons. So inspecting this network lets us identify potential points of coordination between the regulators. For example, HSPA4 here is a hub that connects mRNA level regulators with protein level regulators. And so studying these can give us some useful information about what further experiments we can do. So just to summarize, our integrated network enables interpretation of the host response data as well as guides further study. And I'd like to close by thanking my funding agencies, my co-authors, and very much the organizers of this conference. Thank you.
So, before you brought in the physical interactions, did you look at if there was an over-enrichment for your predicted regulators, if there were like transcription factors?
Yeah, so at the expression level, they were required to be transcription factors or signaling proteins. At the protein level, they weren't. And we didn't really see we didn't see transcription factors from the protein level regulators. We saw more like higher level regulatory proteins like APP and ISG15, where ISG15 is a modifier, actually.
But before you brought in the physical information,
yeah.
If you didn't have that restriction, would you have de novo over-enriched for the signaling proteins and the transcription factor binding data? Did you explore that?
Sorry, so do you mean like, do our modules represent known targets of transcription factors, or do our regulators represent signaling proteins and transcription factors?
Yes, so if you took an unbiased approach and then discovered what are the regulators,
would
they over-enrich for?
So I don't know what MERLIN would do, yeah, because we give it a candidate list, right? So without that, I'm not quite sure what it would do.
Okay, thank you.
Thanks.